Hello and assalamualaikum to everyone who are here for watching this video. I would like to say thank you to all of you and especially to Dr. Shikin for making this video possible. In this video, me and my friend Mira, Haris and Sue represent to you on this particular research titled as The Effect of Cell Destruction Method on the Extractions of Bioactive Metabolism from the Microalgal Biomass.
As for the introduction, biomass from microalgal has been highly demanding widely nowadays. It is a unicellular aquatic plant-like organism that can be found living in a freshwater or saltwater environment that undergoes photosynthesis. It has caused a great contribution to phytoremediation, pharmaceuticals, and other fields to improve modern society, such as the production of biofuel. For example, Syndesmus acanthus and cholera are suitable strands to be used for various purposes in biotechnology application, as Brennan and Amanda on 2010 mentioned that both are prime candidates for mass culture due to their relatively fast growth rates, high lipid content, and the robust nature of the cell. The previous study by Arama on 2014 highlighted the potential biological activities of 5 Syndesmus strands and 3 cholera strands, including antioxidant, antimicrobial, and acetylcholinesterase inhibitory activity. Hence, in this study, we are focused on the extraction of antioxidant and plant biostimulating compounds in both strands. This issue can be overcome by proposed methods such as freeze-drying combined with sonication or ball milling to lysis the cell wall, while mung bead routing bioassay technique is used for extraction of plant biostimulating compounds for biological activity outcome in both strands. Even though microalgae are easily accessed as it can be found at water sources, there are few challenges in conducting the study, such as the issue of energy efficient and cost-effective harvesting. To suitably apply the method on an industrial scale, the objective of this study is to investigate the effect of freeze-drying combined with sonication or ball milling cell disruption method on the extraction of antioxidant and plant biostimulating compounds in cholera and Syndesmus acanthus strands, besides to examine and compare the efficiency cell-disrupting methods and to enhance the extract microalgae cells by using mung-bead assays. We hypothesized that the use of those cell disruption methods studies have given effect on the extraction of bioactive metabolites in cholera and Syndesmus acanthus.
In this section, I will be presenting the taxonomy classification of the microorganism that we will be using in the research proposal. The first microorganism that we will be using is cholera sp. So here, we have 8 levels in the hierarchy from the kingdom to the genus. Next, we have the taxonomy classification of Synedesmus acanthus. In here, we have 9 levels in the hierarchy from the kingdom to the genus Synedesmus. Next, I will be presenting about the method that we will be using in this research. The two methods are freeze-drying combined with sonification and freeze-drying with ball milling. So, what are the freeze-drying? Freeze-drying is a water removal process typically used to preserve a material that easily be destroyed or to extend the shelf life, or to make it easier for the transportation. While sonification is a process that applies the sound energy to agitate the particle in the sample for various purposes, such as the extraction of multiple compounds from plant, microalgae, and seaweeds. Next, I will be talking about the ball milling. So what is ball milling? Ball milling is a crushing technique that grinds nanotube into amazingly fine powders. During the ball processing process, the impact between the little and flexible ball in a disguised compartment will be produced limited high weight, based on Lynch 2005. However, ball processing is anything but difficult to work and reasonable for powder, polymer, or monomer process initiated harm on the nanotube can have.
Let's take a look on the first part, which is simplified as the cultivations of microalgae. The 2 microalgae that will be used, as we mentioned before, will be inoculated in 250 ml liquid center 8 mediums, then Grow the culture with a temperature at range 24 to 26 degrees Celsius with the same ratio of light and dark phase. Plus, during the light phase, the culture will be illuminated with 130 rpm and irradiate with 20 liters per hour of 1.5% carbon dioxide and rich sterile air. Now, the growth and macromolecule measurement. Firstly, we will separate the amount of flux contained in the sample into two different groups. The 30 flux will be harvested on different days to monitor the growth. However, the 70 remaining flux will be used for the biochemical extraction and cell destruction method. Next, we are on the cell destruction method, where the sample for the previous steps will be divided into three different portions. Firstly, we have a freeze drying as a control method. Then we have freeze drying with sonication. Lastly, we have freeze drying with ball milling method. For the combination methods, both freeze-dried biomass will be resuspended in 100 ml of the distilled water before undergoing sonication and ball milling procedure. The procedure will take about 3 minutes and 30 minutes respectively, then repeat the freeze-dry method again. The temperature of the extraction during sonication will be increased from 22 to 31 degrees Celsius. However, the ball milling procedure requires constant temperature at 22 degrees Celsius with addition of 15 gram glass per 30 ml of the suspension. After all, the membrane damage detection will be conducted by measuring the electrical conductivity of the sample. So here are the steps involved in the process. The biological activity, where the effect of cell destruction method will be observed by using the mung bean routine bioassays. On the day 8, the stem will be cut and placing with other 2 leaves inside the prepared solution for 6 hours. After that, we rinse and transfer the samples inside the clean vials containing the water. But some will have the additional of distilled water as a control, and another remaining we have the IBA as a positive control. The final step, which is statistic analysis. There are 3 ways of analysis process, which are the one-way ANOVA followed by a post hoc test, calculate the significant difference of the cell growth, conductivity, yield, and the routing activity. Then, we will use one-way ANOVA followed by pairwise home seduct test to calculate the significant difference between protein, carbohydrate, and lipid. Finally, the software names as the general analysis of variance will be used to measure the cell destruction method, microalgae harvest, and the routine extractions concentration.
By the end of the research, the study will expect to develop a reliable approach by measuring the effect of various cell destruction techniques. This will be measured by observing the cell structure, extract yield, antioxidant activity of chlorella and S. acutis. Next, we expect that the method introduced will give an effect to the extraction of bioactive metabolites in chlorella and S. acutis. Lastly, the study will observe the proper experiment design for bioactive metabolites extraction from microalgal biomass. Based on the studies conducted by Wenyi and fellow researchers in 2020, both cell destruction method brought by higher concentrate yields from the biomass concentrated with freeze-dried biomass, utilizing half methanol as a dissolvable. Both cell disturbance technique diminish cancer prevention agent as acutis X3Ks. The research proposal is supported by various references from established sources, including published book, journal, and studies. Thank you.